I am Jay Kim, and today I will present my analysis of the second iGEM project that I chose. The team I chose was the Korea HS, or the Core HS team. The goal was to design a hyperstable antibody with cell-penetrating peptide for intracellular targeting. Their chosen problem was that antibody functioning is only limited to cell surface receptors and not to the inner cell, as their disulfide bonds are broken in reducing environments. The team conducted multiple sets of experiments to find out the method that maximizes antibody hyperstability and functioning, and this is one of the experiments. First, they cut the SCVP-CPP, which is a cell-penetrating peptide, from the PUCIDT gene. Then they cut the PET-28A vector and inserted the cut-out CPP during a process called ligation, which is the joining of two DNA strands or other molecules. In these two processes, NCOI and SAL-I restriction enzymes were used. Next, agarose gel was made for DNA electrophoresis, which is a common lab technique to identify, quantify, and purify nucleic acid fragments. Lastly, they checked the combined vector through DNA gel electrophoresis and analyzed the results. The team also conducted many other experiments using the peptide, such as transforming and expressing the recombinant plasmid to E. coli competent cells. Now I will present our proposed project from the Synarmos World Academy-wide team. Firstly, our goal was to effectively prevent rusting with the use of magnetotactic bacteria. The problem was that rusting causes metals to dissolve, get weaker, and cause industrial and safety problems on machines like chains and elevators. Our final product was a magnetotactic bacteria solution that attracts metal particles and repels or shuts out oxygen and senses magnetic fields, so that it will effectively prevent rusting by repelling oxygen from metals. A similarity between the Core HS team and our team is that the Core HS team aims to create a more effective antibody that targets the inner cell, which is a more intensified way to treat diseases compared to the original antibodies. And our project aims to efficiently and more effectively prevent and stop rusting for a long time period compared to short-term solutions such as painting and applying oil. In conclusion, we both aim to create a more effective and efficient way that not only targets short-term period solutions or surface solutions, but intensify and deepen the effect of the target solutions. Now I will compare and contrast the system level, device level, and parts level of the two teams. Firstly, the Core HS team used a single-chain variable fragment, or the SCFV peptide, while we used magnetotactic bacteria. Second, in the device level, the Core HS team used NCOI and SAL-I restriction enzymes to cut the peptides and vector and culturing tools in their further experiments, while our team used an oxygen-repelling device and a magnetism detector. Lastly, in the parts level, the Core HS team used a cell-penetrating peptide, a single-chain variable fragment, and a single-chain variable fragment with CPP, while our project used promoters that will regulate gene expressions and functions. These are the sources that I used. Thank you!